My name is Dr. Mauthusi Mitra, and I am currently an associate professor in the Department of Biology, and I work on molecular photosynthesis using a green alga. And here at UWG, I created a research project that will last me lifetime, which focuses on characterization and identification and characterization of novel genes. My lab over the past 6 years at West Georgia have created a mutant library in this algae. And this library we generated to find out novel mutants that cannot grow under high light. You take a foreign piece of bacterial DNA and you throw it into the algae cell. And you do not know where this foreign piece of DNA will go and insert itself in the chromosome of that algae. And wherever it gets inserted, it knocks out genes. And that's how you generate the mutant. And that's why it's a random process. We call it random insertional mutagenesis. So we identified 20 novel mutants, and each mutant will, characterization involves a lot of molecular work. And so currently the ORSP grant focuses on 3 of this novel photosynthetic highlight sensitive mutant. And our preliminary data from one of these 3 mutant is very exciting, because we have identified a novel gene for the first time, and we are thinking that this is involved in photoprotection in this algae to help them grow under high light better. This alga is very unique compared to green plants. It can grow photosynthetically just like your trees, or it can grow heterotrophically just like a fungus. If you give it vinegar, it will use vinegar as a carbon source and make sugar and won't do photosynthesis. Because of this unique nature, I was able to grow this algae on vinegar-containing plate. But when you do the experiments under high light, you saw that when it's doing photosynthesis, when there is no vinegar in the media, it's not growing. When you have the vinegar, it is struggling, but it is growing a little bit. So this opens up this exciting possibility, this application possibility. So if you can clone this gene and put it back in agriculturally important plants and overexpress these genes to make more protein, the plants can grow better under high light. Particularly for some crop plants, it could be very useful. The second application is with respect to the green energy biotechnology sector. If this gene can be manipulated in these strains that the biofuel industry is interested in, it might open up exciting opportunities for these cells to grow nicely under high light and capture solar energy better and convert it into commercially important product like biofuel. So this gene has never been studied on this planet before, and it is missing in higher plants or any other animal system. So that makes it even more complex, but very, very exciting, because our lab for the first time will give a functional clue or role to this uncharacterized gene.